Okay, we're going to get back to neural processing, and specifically the neural processing that's occurring in the retina and then going beyond the retina. So here's some reminder of the wiring. And I know either you're like really cool, or you're like, I don't understand and I don't like this. The wiring is like this. When we think about the wiring to the brain. So those cones that are in the fovea get a linear circuit. They have a 1-to-1 wiring with about one retinal ganglion, or one foveal cone communicating to one retinal ganglion cell. And what this means is it gets greater or clearer representation in the brain, those particular specific cones, which gives us the high acuity. This is why we point our eyes at what we want to get detailed information from. We have convergence in the periphery, where we have many, many rods or cones communicating to one retinal ganglion cell. It does give us this kind of spatial summation because these are rods and cones are distributed across space back there on the retina. With the rods, they have a convergence of about 120 rods to one retinal ganglion cell. And with the cones, we have a convergence of about 6 cones to one retinal ganglion cell. And that spatial summation, so the more summation that we have, the better our sensitivity. And what I mean by sensitivity is sensitivity to dim light, being able to see when it's dim or dark. But we get a decreased resolution. And I know this is all repeating. And again, if you liked it before, you're like, okay, this is just repeating. And if you didn't, you're still probably, I'm hoping the more you kind of hear it and think about it, the more sense it makes. Wiring before, we saw that the rods and the cones are sending signals through to the bipolar cells, and then through the bipolar cells to the retinal ganglion cells. And that's what we're talking about with this wiring, or how these are wired together as we look at these neural circuits. And so far, we've kept it kind of simple, and I've said, well, you don't need to know yet about these other cells, but they are doing this horizontal or perpendicular communication in comparison to what we've just talked about. So the horizontal cells are really existing between the receptors, and then we also see that between the bipolar cells and retinal ganglion cells, we have these amacrine cells that are, again, communicating perpendicularly. So this is usually discussed as vertical versus horizontal communication, but communication going one way, and then the horizontal and amacrine cells allowing for lateral communication or perpendicular communication. And I am going to go through this wiring diagram one more time because I have asked students before whether they want me to. In almost every class they say, yes, please. There's always somebody saying, yes, please. So if we have 2 lights out there hitting these 2 rods, they're communicating, all those 5 rods are communicating to the one bipolar cell. And if those lights are bright enough, we're going to receive enough information that the bipolar cell is going to fire. And I'll just use the example of 4 units. So if I need 4 units of activation to fire, and each of those rods, those lights is at 2 units, then we get 2 4 units, and the bipolar cell is firing. But we can move those lights, right? We can move those lights so that they're hitting different specific spaces or different rods. And what happens at the level of the bipolar cell is it's still just firing at the same rate, right? It just fires. It doesn't know where those lights are coming from. It can't distinguish between which of the rods are getting stimulated. It just knows that it's getting enough stimulation to fire, because that's how neurons work. If we move this down to the cones, and our bipolar cells need 4 units of activation to fire, and each of those lights down there is 2 units, then I'm hitting 4 and 5, those 2 cones, cone 4 and cone 5, but it's not enough stimulation, right? I need 4 units of activation in order to fire as a bipolar cell, and so that dim light, I just don't see it. I'm not sensitive to that dim light. But we're going to turn those lights up now, so that they are at a higher level. They're a little bit brighter, and we're getting 4 units of activation. At the level of if we're looking at the rods, and at the level of that bipolar cell, it's now getting 8 units of activation. It fires faster. But again, I can move those lights around and it doesn't know exactly where those lights are coming from. It's just firing faster now and knows that the light is brighter. If we move down to the cones now, and cone 4 and cone 5 are being stimulated by 4 units of activation of each cone, and then so each of those bipolar cells is going to fire. Bipolar cell 4 fires and bipolar cell 5 fires. We have these two separate cells firing, and they're going to go and get representation in the brain, and we can see where those lights are, right? Because we're not hearing from 1, 2, and 3 in this diagram. But if I move those lights so that they're hitting cone 3 and cone 5, now bipolar cell 1 doesn't fire, bipolar cell 2 doesn't fire, but bipolar cell 3, it fires. Now bipolar cell 4 is not firing, and bipolar cell 5 is firing because we get enough units of activation that I can see the light, and now I can see that exactly where those lights are, right? And that they're split up in this area where there is no light at bipolar cell 4, so that it's two separate lights, and they are in this particular place, and they are separated from each other. So we're getting that detail, we're getting that resolution to the information that's out there. I am going to go back through these circuits very briefly. I did this in with a bit more detail earlier, but so if we're looking at bipolar cell B and we're looking at this as just what is the firing rate of this bipolar cell, and I have a light out there that's hitting receptor 4 so that we're at a unit. I'm always using these really arbitrary, let's say, units of activation and talking about firing rate, even though it is a bit more tricky because you have to talk about wattage or lumens of light, and then we actually are hyperpolarizing the cell, and I keep glossing over that. You can completely ignore those complications. But so if we have a unit of light hitting receptor 4, bipolar cell B is going to fire at a rate of 1, and I can take that dot of light and make it into a bar hitting 3 through 5. I'm still, if I'm bipolar cell B, I'm only hearing from the cell 4, so I'm still firing at a rate of 1, and I can continue to pull that light farther and farther out into a longer and longer bar, but I'm only hearing from cell 4. So with this simple circuit, hearing from cell 4, I'm going to fire at a rate of 1. If we make this more complex, and that bipolar cell B is actually hearing directly from cells 3 through 5, and then indirectly with this, what I'm going to start talking about now as lateral communication, that horizontal communication that we ignored those horizontal and amacrine cells before. But here, if those horizontal cells are now bringing in information from cells 1 and 2, and bipolar cell C is bringing in information from cells 6 and 7, now what happens when I have that dot of light hitting cell 4? I'm going to fire at a rate of 1. I pull that light out into a bar so that the light is hitting cells 3 through 5, and now bipolar cell B is going to start firing at a rate of 3 as I'm getting more information. Since all of these have excitatory connections with bipolar cell B, the farther I pull that out to hit 1 through 7, now I'm going to fire at a rate of 7, right? Because they're all giving me that excitatory information. But in the eye, and what we're going to see for all of our sensory systems, for any sensory system that would exist in any universe, what we're going to have to see is not just this kind of convergence, but some inhibition as well. And so now bipolar cells A and C have that inhibitory synapse with B. So now if I have a dot of light hitting cell 4, it's exciting B. I'm at a unit of 1. I'm going to fire at a rate of 1. I pull that light out into a bar. It's hitting 3 through 5. And now I'm getting those 3 units, and bipolar cell B is firing faster. I pull that bar of light out even farther, so it hits 2 through 6, or let's just go to 1 through 7, because we're going to make this a little bit more brief. And now that 1 and 2 are communicating excitatorily towards A, but A is sending some amount of inhibition to B. Cells 6 and 7 are giving excitatory information to C, but C is sending some amount of inhibition to B. And so now we're actually slowing B down a bit, right? Because it's getting some lateral inhibition. When we look at the receptive field of a neuron, or what we mean by a neuron's receptive field, for vision, this means the area on the receptors that's going to influence the rate of firing of that neuron. So any receptors, so remember that what's happening at the level of the retina and the receptors is there is a visual world out there, and it's getting, by the optics of the eye, flipped upside down and inverted in such a way that it's hitting the back of the retina upside down, and I'm getting some amount of light hitting my various receptors across the retina. So these receptive fields of neurons, or where neurons are receiving information from, their receptive fields are going to have different sizes, shapes, and properties, and we're going to start by talking about center surround receptive fields. This was one example of a receptive field, and this is where we are in the eye, as well as when we get this to the first level of synapsing in the brain. We learn about these receptive fields well. The people who learned about our receptive fields in the earlier part of processing and all the way up through the visual cortex were David Hubel and Torsten Wiesel. And they learned about it by utilizing cats, and they would basically anesthetize the eye of the cat so that the eye is very still. And then they would show or shine a light. This is showing kind of a shadow at different places out on a screen, and they would measure from a single optic nerve fiber. So remember, the optic nerve is a cable of those retinal ganglion cells, axons. There's about 1 million of them in there. They're going to go in and record from just one of those axons, or one of the optic nerve fibers. And as they're recording from that axon, they're going to show things out. So up at A, and remember, because of the optics of the eye, if I'm up at A out in the world, I'm down at A prime on the eye. If I'm at B out in the world, it's hitting at B prime. And if I'm down at C in the world, I'm up. I'm hitting the retina up at C prime. When we look at what's happening at the retinal ganglion cells in these axons, what they found was, so if they shine a light somewhere in A, in that outer surrounding part, we're going to look at the top graph. And remember, we went from negative 70 millivolts up to positive 40 millivolts and back down to negative 70 millivolts, or we overshot. That was the action potential. So we're measuring these action potentials. And what you can see in A, that I like a few things about this. It shows the spontaneous firing. Even before the light comes on, there is this fire, right? I turn the light on, and we still see the same spontaneous firing rate. I am not influencing that neuron, and I am not part of that neuron, that particular neuron's receptive field, if I'm out somewhere in A. If they shine a light in the center there at B, when the light we see at B, there's a spontaneous rate of firing. We see the neuron fire. Turn the light on, and the neuron starts to go fire, 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 fire. Right? It's firing more quickly because it's responding to the light, and it's an excitatory kind of firing. So we're going to put that as a positive or excitatory region influencing that neuron. And if they shined a light somewhere, and so it hit in that outer concentric circle of C, outside of B, but inside of C, okay, we see the spontaneous firing at C in that bottom graph. Turn the light on, and the neuron goes quiet. Turn the light off, and there's this fire, fire, fire. There's this rebound. That's what happens when we inhibit a neuron, it then rebounds. And so this is the inhibitory space. So we have this excitatory center, inhibitory surround, receptive field. All of that region within C is influencing the firing rate of the neuron, some of it excitatorily in the center, some of it inhibitorily in the more surrounding area. What this means when we're looking at dot of light, or I had it as a bar of light before, but we'll use a dot of light and we'll just make that grow, that if I'm hitting that center, I turn on the light and I'm going to see this fire, 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 fire. I make that ball of light larger, so we're looking now in the picture in B. I make that ball of light larger so it's hitting the whole center, excitatory center. It fires even faster. I make that ball of light even larger so it starts to hit that inhibitory surround, and I slow it down a bit. I make that ball of light even larger so that it hits the entire surround, and I slow that neuron down even more. So that is the example of an excitatory center inhibitory surround, center surround receptive field for a particular neuron. What it's going to respond best to is small dots of light. And then as we get too big, it's going to respond less to that. So we're looking at excitatory center, inhibitory surround, but these center surround receptive fields, they can be inhibitory center, excitatory surround. How do we know about inhibition, inhibitory processes in the retina? Well, Hartline and colleagues back in the 1950s were doing research on the limulus, or the horseshoe crab, and they have a much simpler visual system than we do. If you've been to the South Carolina beaches, you've seen these little guys. I'm sure there are other beaches as well, but that's where I've spent most of my beach time. So you've seen these little guys. Sometimes they're stuck upside down and they're dead. But so if we look at what's happening at their visual system, we're looking at the picture on the right now where there are some receptors, and then you can see this lateral plexus that's connecting those receptors. And if we shine a light just at A, or what they did was they shined a light just at A, and then they recorded from that axon of that neuron. These guys have pretty large neurons, so they were easier to do the single cell kind of recording from. And what they found was shining a light at A made that neuron fire faster. And then they shined a light over at B as well, and we could think of this as a single light. And what they found was now with A and B, both of those lights coming on, it didn't fire as quickly as when there was just a light hitting A. And if we look at our figure in C, at the bottom of the figure, now I'm shining more light at B, right? It's hitting a few of those receptors. So even more of that inhibition is affecting the axon of A. We're sending even more inhibition, so I slow it down even more. And that inhibition in the horseshoe crab, that's being sent along that lateral plexus. Okay, so I'm going to respond better if there's just a point of light over at A, at this particular neuron, in this particular axon. Our eyes, human eyes, are much more complicated than the limulus. And instead of having receptors that are just communicating to the brain and then this lateral plexus, what we have is receptors that are communicating to the brain through bipolar cells and through retinal ganglion cells. We're already getting quite a bit of processing right there in the eye. Hasn't even gotten to the brain yet, right? And we have those horizontal and endocrine cells. Those are what allow it, they are basically acting like the lateral plexus in a much more kind of complicated way because we have these various levels. They're allowing for that lateral inhibition. And we are seeing this convergence of these 126 million photoreceptors, with again the rods doing this slightly differently than the cones because there are so many more of them, and because the foveal cones have a linear circuit, but then there's inhibition along with that convergence. So we've gone from 126 million photoreceptors to 1 million retinal ganglion cells. They're sending their axons to the brain. They're making up the optic nerve.